Let's now look into the actual process of going from genetic information to a protein. In eukaryotic cells, DNA resides within the nucleus. So how do we get that information out to where protein products are made? And that happens outside of the nucleus. To do that, we have to transcribe the information that's on a chromosome onto something called messenger RNA. This is kind of like a short snippet of information. Once that information is transcribed on messenger RNA, that messenger RNA can then leave the nuclear envelope through a nuclear pore, and outside of the nucleus, but within the cytoplasm of the cell, that messenger RNA will then undergo the process of translation, where a product is then made, something like a polypeptide and a protein. We'll go through that process in more detail, but here it's really important that you understand what's going on. Where is information jumping from one form to another, and in what part of the cell is this happening? So another way to think of this process is that the DNA within the nuclear envelope is kind of like the library. It has all of the information that your body needs in order to grow and to reproduce and to make protein products. Well, that information is not allowed to leave the nucleus. It's stuck in that, we'll call it a library. That library is in the nucleus. You can't take the books out. So what do you do when you can't take books out of a library? Well, you take notes. You take notes on those books and you can leave with your notes. So the process of transcription is kind of like the writing down of the notes. It's the process of writing the notes and copying them down. When you're done writing your notes, you're left with whatever information you wrote down, and we'll say that that's kind of like plans. It's plans for a protein, and that's your messenger RNA. Those are the actual notes. So transcription is like the process of writing the notes, and the messenger RNA is kind of like the notes themselves, the information that you've copied down. And in this example, the notes are the plans to make an airplane. We'll say that there's notes that have been made and they've been written down on how to make this airplane. So then these notes can then leave the nucleus, and once they're outside of the nucleus in the cytoplasm of the cell, then translation can occur. And translation is kind of like taking those notes, taking that information on how to build something and actually building it. The end product are those polypeptides, are those proteins, and in this analogy, it's like the actual physical airplane that you made from the notes that were copied down inside the library. First, let's look at transcription, that initial movement of information from DNA to messenger RNA. This diagram illustrates the process of elongation, which is a stage of transcription. During elongation, RNA polymerase, which is an enzyme, it moves along the DNA template and adds nucleotides by base pairing with the DNA template in a way that's really similar to DNA replication. The difference is that in this case, an RNA strand is being made, and that RNA strand, it doesn't attach to the DNA. It remains free, and that will be the messenger RNA. Once that full messenger RNA strand is transcribed, the RNA polymerase will disassociate from the DNA once it hits a certain code, a certain line of repeated code that signals to the RNA polymerase to disassociate from the strand of DNA. Let's look at this process in a little more detail. So here we have a gene. That's a certain region on a strand of DNA that codes for a particular product. On that gene, we have a promoter region and a terminator region. The promoter region is where this synthesis of the messenger RNA is going to begin, and the terminator region is where it's going to stop. So RNA polymerase approaches the promoter region, and it associates with the strand of DNA. Within the RNA polymerase, the strand is separated, it's unzipped, however you want to think about it. It's basically separated in a way where it can now be copied. Different base pairs are assembled of RNA, and that messenger RNA strand gets longer and longer. As the RNA polymerase moves along, it creates that strand of messenger RNA. You can see it there at the bottom. And eventually it reaches a terminator region where it disassociates from the strand of DNA. Then we're left with a strand of messenger RNA. And remember that this strand of messenger RNA is a gene. This is all the information needed for a particular product. And that product is a polypeptide or a protein. It could serve a lot of different functions in the body, but this is basically a unit of information for a product. That RNA undergoes some processing to help make it a little more durable and longer lasting within the environment in the cell. And then it leaves through a nuclear pore. It leaves the nucleus. Remember, this is like the notes leaving the library. Now that transcription has occurred and we've put that information onto messenger RNA, and that messenger RNA has left the nucleus, we can now look at the process of translation, where that information goes from genetic information to a gene product, something like a protein. Here are some triplets. So this is a strand of messenger RNA. It has many different base pairs. And remember, we're reading these base pairs in triplets, in threes. In each group of three is a codon. We have a start codon, AUG. This start codon is where the ribosome will attach and begin the process of constructing that protein with the help of tRNA. Transfer RNA is kind of like a rechargeable building unit. So right here you can see that the transfer RNA has a little sphere at the top, and that is an amino acid. Well, once that transfer RNA gives up that amino acid, it can then go back into the environment and collect another amino acid and be used again later. So the transfer RNA is going to come into play over and over and over again, and each time it comes into play, it will have one of those small amino acids. It will attach it to another amino acid, and then it'll actually leave the ribosome with no amino acid and kind of get recharged somewhere else. Each kind of transfer RNA carries a different amino acid, and the amino acid that it carries is indicated by the triplet at the bottom of the amino acid. So it has a complementary base pair at the bottom of it, and that complementary base pair will match up with the codon. When they match up, that means that the transfer RNA will then give up its amino acid. We call the base pairs at the bottom of the transfer RNA anticodons, and those are complementary to the codons. So in this case, the codon is AUG and the anticodon is UAC. Once the large ribosomal subunit attaches to the top of the small ribosomal subunit, it then directs transfer RNA into place. You can see here that the next tRNA, which is charged, it contains that amino acid on top, is approaching, its anticodon is approaching the complementary codon that's on the messenger RNA. Once it's in place, the two amino acids then join up, and the ribosomal subunit moves over another space. The uncharged tRNA is then released. You can see now that the transfer RNA, the tRNA, now instead of having just one amino acid, it now has two amino acids. The ribosome will then move along the strand of messenger RNA, and transfer RNAs will come in and leave each time, assembling one more amino acid, until finally it makes a whole chain of amino acids. Eventually it'll hit a stop codon, and that will cause the ribosomal subunit to disassociate, and you will be left with a polypeptide. Here's a polypeptide. So you can see that this polypeptide is a string of different amino acids, and we now know how those different individual amino acids were assembled. They were put together by transfer RNA, which was directed by the ribosome. In prokaryotes, the regulation of when to express genes, that happens at transcription. Once that information is transcribed, the information quickly becomes gene products like proteins. In eukaryotes, there's a lot of different places where the expression of a gene can be regulated. It can occur during transcription, in the nucleus, during translation. It can even happen in later stages, which are called post-translational modifications.